Thank you for joining us at the Kirby Institute seminar series, and also to those who are joining us online. My name is Dr. David Van Bockel, and I'm representing the Kirby Institute, and particularly the Immunovirology and Pathogenesis program, excuse me. And I'm joined by my colleague, Bethany Horsberg, as co-chairs of this session. I would like to begin by acknowledging the traditional custodians on the land in which we gather today and pay my respects to elders past and present. Today, we're presenting from the Bidjigal people of the Eora Nation, and I extend that respect to Aboriginals and Torres Strait Islander people who are joining us today here in the audience or who are online. I might just start off with a little bit of housekeeping. The format of the seminar will start off with a presentation, followed by a Q&A session afterwards. If you're joining us online today, you can ask questions throughout by clicking on the Q&A icon at the bottom of the screen. For our audience in the room today, there's a microphone set up available at the side of the room to address any question at the start of the Q&A session and at the start of the Q&A session at the end of the presentation. So right now, I'll get my colleague, Beth Horsberg, to introduce Gabriel for today's seminar.
Thank you, everyone. It's really a great pleasure of mine to introduce my dear friend, Gabby. Gabby completed his doctorate at the University of Buenos Aires in Argentina. Sorry about that pronunciation, Gabby. In 2019, working with HIV immunopathology, particularly working on extracellular vesicles. I then had the joy of meeting him when he did a postdoc with us over at WIMA later in that year, where he's remained ever since. Gabby's portfolio can best be described as kind of expansive, but with a focus on immunopathology. He works on HIV control during art interruption, HIV maintenance during the reservoir development. During COVID, he worked with UNOC working on proteomics and immunopeptidomics, which is a very big mouthful, but a very important concept. And he also delves into CAR T-cells whenever he has the time. Amongst all of that, he also happens to be one of the most skilled barbecuers that I've ever met in my life. So today, Dr. Duette is going to be talking to us about two of the greatest killers in the world, HIV and TB, and how they play off each other to generate an environment that can prevent HIV persistence. So go for it, Gabby.
So hello, everyone. I would like to thank the kind introduction by Beth. We all miss you in WIMA. And I would like to start by acknowledging the traditional owners of this land, the elders past and present. Okay, so first, I would like to thank Samantha Cronin, who is the PhD student leading this project, and also to Sarah, who is the person who's supporting this project since the beginning. And as Beth said, I will talk a little bit about co-infection between HIV and tuberculosis. And why is this relevant? Basically because people infected with the Mycobacterium tuberculosis, which is the agent for tuberculosis disease, the co-infection with HIV for these people actually can increase the probability of develop the TB or the tuberculosis disease. And this is higher in co-infected people when compared with a person infected just with the Mycobacteria. And even under efficient antiretroviral treatment, the chances of developing active tuberculosis is higher in this population. People infected with HIV, also tuberculosis represents the leading cause of AIDS-related death. In our project, we are investigating the effect of tuberculous pleural effusion. And why is this? Because this is a very relevant and physiological fluid, but basically, I will tell you a little bit about how this is produced. When the individual is infected with the Mycobacteria, in most of the cases, these individuals can contain the infection and generate the granuloma. So basically, the immune system can contain the bacteria in a latent state in these granulomas. But for different reasons, such as immunodeficiency induced by HIV, for example, the immune system cannot control the bacteria anymore, and the latent infection pass to be an active infection, and the granuloma can be disrupted, and this release immune components, not just the bacteria, also immune components, and can generate an inflammatory fluid. And this fluid can go over the pleural space. And as I mentioned, this fluid is represented by immune cells, immunomodulatory factors, soluble factors, and some bacterial component as well. So this is why this is a very physiological relevant fluid, but also because up to 30% of the TB patients develop these pleural effusions, and it's more frequent, even more frequent in co-infected individuals. And very importantly, it was found that the HIV viral load is higher in the pleural fluid when compared with plasma from the same donor. And we use this fluid as a model to represent the TB-associated microenvironment and how this microenvironment can impact the HIV infection. And this model has been used for our collaborator in several works to study the co-infection, but also how this microenvironment can enhance the infectivity or the transmission of Mycobacterium tuberculosis. So if you go to the literature, you will find a lot of work about how HIV can impact the infection with the Mycobacteria. And most of these work are focused on the macrophages because this is the common target between both pathogens. However, little is known about how the Mycobacteria can actually impact the infectivity of HIV. And for example, our collaborators have published this beautiful work where they see that this microenvironment actually can enhance the spread of HIV among macrophages. And this is because this microenvironment, basically IL-10 in this microenvironment can induce the generation of tunneling nanotubes, and that promotes the spread of HIV in these cells. However, as you know, one of the main targets of HIV are the CD4 T-cells. But again, little is known about how this environment can impact the infectivity of HIV on the CD4 T-cells. So the main aim of our study is to investigate what is the interaction of HIV and CD4 T-cells in the context of the co-infection. And our hypothesis is that this microenvironment can enhance the infectivity of HIV in CD4 T-cells. So we developed a collaboration with a group in Argentina. This group is led by Luciana Balboa. Dr. Luciana Balboa get very valuable samples from co-infected individuals, and she obtained the pleural fluid from these individuals and blood. So we could quantify the viral load in both fluids. And interestingly, what we see is that when we compare both compartments of both fluids, the higher viral load is much higher in pleural effusion when compared with plasma from the same donor. So as I mentioned, this was already reported, but was in just one cohort. So I think that it's important to show that this can be reproduced in a different cohort of participants. And in our lab, Katie Fisher, a brilliant postdoc, has developed a sequencing assay to sequence near full-length RNA HIV. And by using this technology, we sequence the virus from pleural effusion and plasma from one participant. This is ongoing. We want to increase the number of samples, but at the moment, this is what we have. But we think that it's relevant to share with you what we have so far. So basically, Samantha and Katie, they sequence the virus, and what we can see after phylogenetic analysis is that we cannot see evidence of compartmentalization between both fluids, and meaning that the pleural space is not isolated from circulation. And we haven't found identical sequences between both compartments, but as you can see here, they are genetically close and related. This is ongoing, it's preliminary, but Samantha also sequenced the provirus from mononuclear cells, from pleural effusions, from these co-infected donors, and from blood from the same donors. And again, this is preliminary. So far, we have the data from one participant, but we are expanding this to additional donors. So what Samantha found, and we are sequencing the provirus from these infected cells because we can get a lot of information from this sequence analysis. And you are probably familiar with this concept, but in infected cells with HIV has a provirus integrated in the host genome. But this viral genome actually can be or genetically intact, able to produce infectious virus, but also can contain a lot of different mutations. And actually, what we usually see in people living with HIV, when we sequence the virus from these individuals, most of these sequences are genetically defective, and they contain large deletion, most of them. So when Samantha analyzed the sequence, and Andrea, she also helped with this sequence analysis. And what they observed is that when they align the sequence coming from PBMCs or cells from blood, around 28% of these sequences are genetically intact. And as you can see, most of them contain big deletions as expected from infected donors. But very importantly and interestingly, what we also observed is that when Samantha analyzed the sequence coming from pleural effusion, cells coming from pleural effusion, the proportion of genetically intact HIV in this cell is higher, up to 46% in these samples. And Yunok Lee, another brilliant postdoc in our lab, she developed an algorithm to calculate the infection frequency based on this sequence data. And what we see is not just the proportion of genetically intact HIV, which is higher, but also the total infection frequency, meaning that more cells are infected with HIV. And as you compare the proportion I mentioned before, you also will find more genetically intact HIV per million of cells. We wanted to analyze the phylogenetic relationship between these compartments in order to see if we find evidence of compartmentalization or not. And as you can see here, this preliminary analysis is showing that these sequences are genetically close. I mean, they are related. We haven't seen evidence of compartmentalization, suggesting that maybe there is some traffic between both compartments. So we see higher viral load, more infected cells, and as you remember, our hypothesis was that this microenvironment can induce or promote the infection of HIV in CD4 T-cells. So we wondered if we can explain why we see this higher proportion of infected cellular virus in the pleural space. And to address that question, we ran a simple experiment. Basically, what we done is to isolate CD4 T-cells from Buffy coat from healthy donors. As you know, these cells have to be activated in order to get infected. And we also use pleural fluid from TB-infected donors. But here I would like to make clear that these samples are coming from people with tuberculosis, not co-infected with HIV, because the system is a bit more clear in this way, more clean, sorry, because otherwise you will have the virus from the fluid, and it's really hard to actually basically take out all the virus from the sample. So in this experiment, as I mentioned, we isolated CD4, we activated them to make them susceptible to the infection in the presence or absence of TBPE. And after 3 days post-infection, we measure P24, which is a protein, viral protein that we use as a readout to determine and quantify the proportion of infected cells. So what we see, and obviously we check that the viability of these cells was not affected by the treatment, and you can see that basically we haven't seen difference in terms of viability across the conditions. And surprisingly, as you can see here, and unexpectedly, what we see is that this treatment actually decreased the infection of HIV in CD4 T cells. And this was consistent across several donors. And to prove that, something I forgot to mention is that for this experiment, we use a pool of these samples. Basically, we have a pool of different samples from different donors, and we treat them to the cells. To prove that it's not a pool effect, basically, we also assess the same experiment, but using individual samples from single individuals with tuberculosis, and we could reproduce the same result that we observed using the pool. And in addition, as a fluid control, we used pleural fluid coming from individuals undergoing heart failure, and we haven't seen exactly the same effect. There is some there, but it's stronger when we use pleural fluid from tuberculosis patients. So from now on, forget everything I said about higher viral load, higher proportion of infected cells, otherwise will be a bit messy. So we try to understand what part of this HIV replication cycle was affected by TBP. So we started with the first step, which is the viral entry. And for this experiment, we use a very elegant system. I'm not sure if you are familiar with this, but briefly, the system relies on the staining of the cells with CCF2, which is a fluorescent dye. And when these cells are infected with a virus containing PPR, which is a viral protein, which is linked to a beta-lactamase, this beta-lactamase can cleave the dye and the fluorescence of the dye shift. And this shift can be detected by cytometry, and we can quantify the viral entry by using this assay. So as you can see here, this is the mock condition, and with HIV, we can detect this shift I mentioned, so we can efficiently detect the viral uptake from these cells, but we haven't seen difference in the viral entry when we use TBP or we treat the cells with TBP. So then we analyze the next step, which is the reverse transcription, and we did that by qPCR. As you know, the HIV genome is RNA, and by the viral enzyme can reverse transcribe to DNA. And this step can be quantified by qPCR by using primers that basically can detect the early and late DNA product of the reverse transcription. And very interestingly, what you can see here is that actually this microenvironment or the treatment with TBP actually drastically can inhibit the reverse transcription step. So we wanted to understand, or we wanted to identify the mechanism behind this effect. And as you know, early events of HIV replication are dependent of the T-cell activation of the cells. And so we perform RNA-seq from CD40 cells activated with anti-CD3 and 28. But basically, we have these conditions. We have untreated cells. We have untreated cells, but in the presence of TBP. And on the other hand, we have cells treated with these antibodies, which will activate the cell through the TCR, and activated cells in the presence of TBP. If you see this PCA analysis, basically, there is a huge separation between untreated cells or unactivated cells versus activated cells. It's not very clear in terms of untreated in the presence of TBP or unactivated, sorry, in the presence of TBP, but we can see clustering patterns when activated cells are exposed to the TBP fluid. So then we ran a differential gene expression analysis, and you can see that actually even in the absence of activation, there are genes that are differentially expressed. And this was also confirmed by a volcano plot analysis, as you can see here. And as I mentioned before, one very important step or something that actually relies the cells to be infected by HIV is the activation level of these cells. And we specifically analyze if the T-cell activation pathway were affected by this microenvironment. And in fact, what we see is that this fluid is immunosuppressive in this sense, can basically downmodulate the TCR signaling and downmodulate the activation of CD4 T cells. And then we confirm this data by staining the cells for CD69, CD25, and HLA-DR, which are activation markers, and we confirm this by flow cytometry. And again, as control, we we assess this activation assay by using individual sample from single TB donors, and also we use as a control heart failure PE. So to make this a little bit more complicated, as you know, early steps of HIV, or basically the metabolic state of the cells, also can impact in the early steps of the HIV replication, and was at this point it's very well known that, for example, HIV needs oxphos and glycolysis to efficiently replicate, and this was proved by several groups. And so we decide to analyze if the metabolism of this cell is also impacted by TBPE. And just a reminder that metabolism and T-cell activation are very interconnected. So, and this was done by our collaborators in France at the IPBS. And Zoe, the postdoc who ran this experiment, basically she activated cells with anti-CD3 and 28, and in the presence or absence of TBPE, and then she measured by seahorse technology, which is the gold standard to measure metabolism. She measured glycolysis rate and oxphos rate, and this is the graph that you get from the seahorse analysis. And as expected at this point, the cell exposed to TBPE, they showed a lower rate of glycolysis and oxphos, but wasn't the same when they compare cells treated with heart failure PE, again, as a control, a fluid control for our experiments. And so basically, if we recap, this microenvironment can decrease the activation of these cells and also the metabolism, in particular, glycolysis and oxphos, which both activation and metabolism are very important for the very early steps of HIV replication. And in our group, we also reported that HIF-1 alpha is a transcription factor which regulates metabolism, cellular metabolism, but also is necessary for HIV replication, was down-modulated by TBPE when they were treated with this fluid. So several components or several aspects that are necessary for efficient HIV replication were down-modulated by TBPE. So far I've been talking about HIV replication, but as you know, a very important aspect of HIV pathogenesis is HIV latency, because you know that latently infected cells, they compound the HIV reservoir. So we also wanted to investigate what is the impact of this microenvironment in terms of HIV latency. And by using our RNA-seq data, we particularly analyzed relevant pathways necessary for efficient HIV transcription or necessary for HIV reactivation. And what we observed is that important pathways for HIV transcription and reactivation were down-modulated, pathways such as NF-κB, interferon pathway, and the JAK-STAT pathway. And importantly, we also see that the TGF pathway, which is reported to promote latency or support latency, was upregulated. So this is indicating that this microenvironment actually could induce HIV latency in these CD4 cells. So to assess this idea, we use a dual reporter HIV virus, which basically contain two different reporters, GFP and IRSP. And the GFP reporter is the expression of this GFP reporter is dependent of the viral LTR. So if we have any productive infection, the cell will express GFP. And on the other hand, we also have the RFP, which has its own reporter, meaning that if the cell is infected, to express this reporter regardless if the infection is productive or latent. So here is an example. You can see cells single positive for RFP. So they are the latently infected cells. And then we have the double positive cells, which are the productively infected cells. So first we infected Jurkat cells. These are CD4 T-cell lines. Is more convenient because these cells are easily infected with HIV. So we started with this model. And as you can see here, the proportion of latently infected cells is higher in the presence of TBPE. And if we quantify the ratio between latent and productively infected cells, we observe that this ratio is higher in favor of the latently infected cells. And then we validated this using primary CD4 T cells, and basically we reproduce the same result, different values, but basically the ratio between latent and productive is always higher in favor of the latently infected cells. So this microenvironment is inducing latency. And as we see this induction of latency, we also were curious about if the reactivation of HIV is impaired as well by different latency reversing agents. So first we started with this model. This model are JLAT cells. Probably you are familiar with this, but basically they are cells infected with one copy of HIV. They are latently infected with HIV, and this virus can express GFP. So basically you reactivate these cells using LRAs, such as PMA, which is a very strong LRA. We can quantify the level of reactivation by measuring GFP by flow cytometry. So for this experiment, we tried different LRAs. We started with PMA, which is the classic and the strongest one, but we also tried P005, which is a PKC activator, the same with PMA. And also we tried GQ1, and GQ1 reactivate HIV for a different mechanism. But basically what we see is that TBPE was able to inhibit the reactivation of HIV for these three different LRAs. But when we tried different LRAs such as TNF or borinostat, these LRAs were not inhibited by TBPE, meaning that there is the effect of TBPE can be pathway specific. It's not basically shutting down every single pathway that is involved in HIV reactivation. And importantly, we validated these results by using cells from HIV-infected donors. And for this experiment, we use PMA, which is the strongest LRA for this kind of experiment. And we see in 3 donors that, again, the treatment with TBPE was able to inhibit the reactivation of HIV in these samples. So all the pool of data we have so far indicates that this microenvironment can inhibit HIV replication and promote HIV latency in CD4 T cells. But as I mentioned before, this contrasts with our first result where we see higher viral load and a higher proportion of infected cells. So we are wondering why we see these contrasting results. And we think that maybe in part can be because the CD8 T-cell mediated clearance of HIV-infected cells maybe is impaired by this microenvironment, in particular because, as I showed before, this environment seems to be immunosuppressive. So to address this hypothesis, we obtained cells from people living with HIV, and we stimulated the CD8 T cells using peptides of HIV to induce an antiviral response and the expression of antiviral cytokines such as TNF and interferon gamma. And also we measured the granulation by staining for CD107. And what we observed is that in the presence of TBPE, the production of these effector cytokines and the granulation were demodulated by this microenvironment. And again, we use heart failure PE as a fluid control, and we haven't seen the same effect for this experiment. So we would like to know what is the mechanism behind in the CD8 sites, and to, as you know, the access to sample from HIV-infected donors is very limited. So we try to undergo or investigate the mechanism by using cells from healthy donors. But before, we wanted to validate if this can be reproduced using these cells. The same we see to cells from HIV-infected donors can be reproduced using cells from healthy donors. So for this experiment, we use a pool of peptide for CMV, VV, and flu. We call this pool of peptide CEF. And similar to what we see with the cells coming from infected donors, HIV-infected donors, we see that the production of effector cytokines and the granulation was impaired when we stimulate the cells with these peptides. And so having validating this, we then we decide to perform RNA-seq analysis again, and this time we also use anti-CD3 and 28 antibodies to stimulate the TCR and activate these cells, and to evaluate if the effector functionality of the CD8 was affected by this TB microenvironment. So what we see is that effector pathways involved for CD8 T cells, they were down-modulated in agreement with our idea. And again, we validated this by measuring activation markers in these samples by flow cytometry. So again, we see this immunosuppressive effect of TBP on these cells. And as I mentioned before, the metabolism, in particular glycolysis and OXPHOS, are necessary not also for replication of HIV in CD40 cells, but also for the efficient effector functionality of the CD8 T cells. So basically, the activation of the TCR pathway can trigger an increase in glycolysis and OXPHOS. And this is also regulated by HIF-1 alpha. So again, employing the seahorse assay, what we see is that when soy treat the cells with TBPE, the metabolism of these cells is lower in terms of glycolysis and OXPHOS, and the expression of HIF-1 alpha was also down-modulated. So finally, we wanted to determine if the killing capacity of this cell was impacted by this microenvironment. And for this experiment, we get back to samples from people living with HIV, and we expanded the CD8 T cells using HIV peptides in the presence or absence of TBPE, and we expand them for 2 weeks. In parallel, we generated the target cells, which are the CD40 cells. So we activate them for also 2 weeks. And at day 11, we infected the CD40 cells, and then we stained them to identify the infected cells by cytometry. I will show that in the next picture. And after 13 days of expansion, we isolated the CD8 T cells, and we co-cultured target cells and effector cells in a ratio 1:3, 1 target, 3 effectors. So you can see here how the cytometry looks like, and we can efficiently discriminate the CD4 population, and on the CD4 population, we quantified the proportion of HIV-infected cells. So this is what you see when you infect your CD40 cells with HIV. And as expected, when we co-culture T cells with CD8 effector T cells, you see an efficient clearance of the infected cells. But when we co-culture these cells with CD8 expanded in the presence of TBPE, we see that actually the killing capacity of this cell is drastically inhibited. And this was consistent across 4 different donors. So in summary, this microenvironment has immunosuppressive properties that decrease the activation and the effector functionality of these cells, but also the clearance mediated by these CD8 T-cells. So as preliminary conclusion, we can say that the coinfection can promote the persistence of HIV by inducing latency of these cells, meaning that these cells are hidden from the immune system, and also impairing the CD8 functionality. And this also is very important because can have consequence in terms of HIV cure strategies, such as shock and kill strategy, which relies on reactivation of HIV and also in the clearance of these infected cells, potentially by CD8 T cells. And so far, a preliminary result indicates that the reactivation of these cells can be achieved for some LRAs, but not for all of them, meaning that maybe for these co-infected individuals, we need to investigate more to determine what treatment can be the best treatment for these persons. So finally, I would like to thank all the members of our group, in particular Samantha and Sarah, and our international collaborators, the funders, and in particular, I would like to acknowledge with gratitude the participants of these studies. Yeah, that's it. I'm happy to take any questions.
Thanks, Gabby. So I'm going to take floor questions. DVB is going to take internet questions. So feel free to ask away either direction. Gabby, just as a first question while we get everyone sort of up and running, probably a little bit of a naive question, but have you thought of the opposite direction? So in terms of, in that microenvironment, you're going to have a lot of things like macrophages and DCs happening. Have you had a look at to see how the transfer is being affected as well? So is it like brute forcing its way as well as causing that depletion?
No, actually, that is a wonderful question because, no, really, because as I mentioned at the very beginning, our collaborators showed that actually this microenvironment promote HIV infection in macrophages, and in particular in the lung, actually myeloid cells take a key role there, and more works are showing that early to say this, but at least there are evidence suggesting that macrophages can be part of the reservoirs of HIV. So actually, we haven't addressed that, but it's very likely based on our collaborator results. So we haven't addressed that, but maybe that partially could explain why there is higher viral load in this compartment.
Yeah, so you get more going in and then more staying in.
Yes, obviously we are thinking a lot about why we see these contrasting results, but also we believe that can be 2 things. Maybe it's the traffic of the infected cells to this compartment, and when the cell get into this microenvironment can block the infection, inducing latency, and in addition, the CD8 killing is impaired. So in summary, you have more virus in the system. But also, we believe that can be a matter of timing. I mean, because we are mimicking or we are reproducing what happened in active tuberculosis and a very advanced stage. And as you know, the immune system tries to shut down the immune response because it can be detrimental. So maybe what we are getting to that point, and before that, maybe the microenvironment can induce HIV replication. So maybe that is why we see this contrasting results.
Hi, thanks. That was a really interesting talk, Gabby. I've just got a question kind of along the same lines as Beth. In terms of the cellular components of the plurifusion fluid, whether you've looked at, I guess, the different subsets, you know, or the immunosuppressive subsets, but also in the cytokine and chemokine content. We've in the past looked at mate cells, and they're really quite predominant in TB in the lung, and actually they make a lot of CCL3, 4, and 5, which all can block CCR5. So I don't know if you've looked at that.
No, not yet, but actually something we are planning to do because we want to characterize in depth what is in this pleural fluid for sure, because we want to determine what is the mechanism behind of what we see. So our first candidate was IL-10, not just because it's immunosuppressive, also because our collaborators identified IL-10 as the main factor driving what they see, which is the promotion of HIV infection in macrophages. But we ran some preliminary experiment showing that it's not IL-10 in our system. So that is why we want to do something more in depth. But yes, we are planning to do that. And in terms of the cell composition, so far we haven't characterized the cells. We use most of them for sequencing. We know that most of these cells in this pleural fluid are lymphocytes and T cells, just a minor fraction are myeloid cells. But we are planning to do 20, I don't remember how many colors, but a big panel to characterize the cells in that compartment. So that is ongoing, and hopefully we'll get some results soon. Yeah.
Thanks very much. Fantastic talk. It really does kind of highlight a pertinent question, not only in the developing world, but also in the developed. Yeah. Like, for example, people who are migrating, out of like densely populated areas with TB and moving into and migrating to like more modern centers like England and the UK and the US where they might seek treatment. I guess the really exciting question would be like what's actually happening in the lung, and have you got any information on like the activation status of the CD8 T cells in there, or are there any kind of like papers that might allude to kind of what's happening there that might give you an indication of the higher viral load?
No, wonderful question. Actually, as I mentioned, we are planning to characterize in depth this cell from this compartment that includes markers for activation, exhaustion. So we want to really characterize further what is happening with the CD8. No, because our system, we are aware that it's a bit is in vitro, but we want to see if that can be reproduced when we characterize the cell from the site of the coinfection. And also we got samples from co-infected monkeys, lung samples. So we are planning to do some microscopy to characterize the T-cell population there. And again, trying to assess for some functional markers like exhaustion and activation, the granulation, and see that we can reproduce what we are suggesting here. If the microenvironment in an in vivo setting actually is also affected by the co-infection.
Great, thank you. So if what you say rings true, and you do think that a lot of what it is you're observing in terms of suppression of HIV actually stems from inhibition of CD8 T cell function, what kind of therapies do you think you might like to try? There's a raft of them out there. Like, what would you be interested in doing?
No, honestly, I don't have the answer now, but at some point I was involved in a CAR T-cell project, and something, this is just a crazy idea maybe, but basically our results suggest that the TCR pathway is impaired. You activate the TCR of these cells, basically they will not develop an efficient response product of the inhibition of the TCR pathway, maybe by the PKC inhibition. So something we thought in the past, maybe we could resume that, is using T-cell therapy independent of the TCR. I mean, for example, if you have CAR T-cells targeting a specific antigen of HIV, and if that stimulation is not dependent of the TCR, maybe that can overcome the challenge of the inhibition of the TCR pathway.
There's a lot of great lessons that could be learned, not only for TB, but there's other like like pleural diseases and sepsis and whatnot that could learn from situations like this. So thanks very much.
No, thank you.
Unless there's any other kind of burning questions in the room, no. Just leaves me to thank Gabriel, and thanks very much for a really wonderful talk. Thank you.
Thank you for the invitation.